Welcome again. My name is Raisa Mäkipää, and we start the second HoliSoils webinar. And today's topic is how biochemistry may support microbiology. And we have a guest speaker, Adamczyk, from Natural Resources Institute, Finland, Luke. And this seminar continues, and further information about project and this webinar series you can find from our website from holisoils.eu. I don't spend more time for introducing the project, but if you are interested in content, you can contact us. We go directly to the topic of the day, which is which I already told, and the floor is yours, Bartosz.
Thank you, Raisa. Hi, guys. So you know already my name and the topic, so we can go to the next slide. So today I will tell you a bit about biochemistry because it is in the title. Later I will show you how biochemistry can help with analysis using DNA and RNA, how complementary it can be. Later I will give you a couple of our new methods which we developed in connection with HoliSoils project. Later I will add something about less known mechanism of carbon stabilization. And last but not least, I will present you our HoliSoils results from nitrogen fertilization experiment as a good case study for using different sets of the methods. Next, please. So what is biochemistry? Yes. As you can see, it's something like a child between organic chemistry and biology. So on the left, you can see biochemistry, but in practice, it's even more complicated. Next, please. It looks more like this. Next, please. So the good definition of biochemistry is just the mechanisms or all chemical processes in living organisms. It includes structural biology, enzymology, metabolism. It is related to molecular biology. So these are just the mechanisms of biological phenomena. Next, please. So first part will be about the methods which we developed in connection to HoliSoils project. There will be something like 4 or 5 methods. Next, please. Yeah. Right now I will at first tell you something more about advantages and disadvantages of DNA, RNA methods, and the same for biochemistry. So for DNA and RNA, using these kind of methods, we can get information about who is there. So for example, microbial community structure is there. We are searching for, I don't know, this or that specific species of the fungi, and with these methods, we can find it there. We can also get information about potential chemistry of microbes and potential to decompose, because if using DNA and supported by RNA, we know that the fungi, this typical fungi is doing typically this or that. Using DNA and RNA methods, we can figure it out. Next, please. In case of biochemistry, we can get more like information about how much of the thing is there. So concentrations of different pools, different markers of different processes. Next, please. So in case of disadvantages a bit of DNA, RNA information would be that it is based more like on about relative abundances, and they are telling about potential to some biochemical processes. Next, please. In case of biochemistry, the disadvantage would be that, okay, we have concentration of the thing A, but we have no idea from where it's coming from. So we do not know who is doing that exactly, but we know that something is going on. Next slide, please. So examples of our biochemical methods include a method for fungal biomass measurement, microbial necromass, arbuscular mycorrhizal biomass. This is under development still. Melanin concentration in case of quality and quantity, and at the end, DNA methylation. Next one, please. And next one. So when we want to know about fungal biomass in the meaning of it's bigger or lower, so we want to have a number of fungal biomass. Using DNA, we can get relative abundances of the species. using qPCR, basically some estimation of the fungal biomass on the basis of DNA. Next one. Using PLFA, we can get something more, but some markers are not so very representative of PLFA. PLFA is phospholipid fatty acids. And it also depends on the phosphorus status, so it depends. In case of ergosterol, which I wanted to underline here, it is a good marker for fungal biomass only because PLFA can also provide us bacterial biomass. But ergosterol is more representative if the correct extraction is applied. And next one. During the HoliSoils project, we figured out, we compared most of the common extraction methods. We chose the most representative, and also we build up a new method which is also including mass spectrometry. So we are 100% sure that it's ergosterol on the basis of mass scans, and also using mass detection we can increase the precision for about 10 times. So we can also quantify very small amount of ergosterol. Next one. So, of course, what I would like to get, it's some biochemical method using which we can tell fungal biomasses of specific functional groups of the fungi. So separately, saprotrophs, arbuscular, ectomycorrhiza, and so on. But it's still not yet available. Next one. But I have one idea here. In case of arbuscular mycorrhiza, these fungi, they are rather not producing ergosterol, but they are producing something very similar. It is 24-methylcholesterol. Next one. So as you can see, using, again, HPLC and mass spectrometry, we can find the differences between these two markers. Ergosterol is the first peak, marked in, let's say, brown. And the second one in 207 nanometers, it is 24-methylcholesterol. I'm still developing the methods. It's not yet ready, but I hope that at the end point, I will be able to provide you with a method which is counting the total biomass of fungi and separately that arbuscular mycorrhizal group. Next one, please. Another method which is already published, we also developed it under HoliSoils. Maybe I will introduce here why to take into account especially microbial necromass. Typically, on the left, we can see that plants are producing a lot of litter, especially from the roots, well, especially from the above ground part of the plants also, but roots are more important for stabilization of carbon. And later, this litter is, microbes are feeding on the litter, they are growing, so they are growing their biomass. Later, of course, they are dying. And from this death, microbes, quite big amount of stable carbon is formed. So now, as you know, why to study microbial necromass. Next, please. To estimate the amount of microbial necromass, you can use two amino sugars, muramic acid, which is marker for bacterial necromass, and glucosamine coming from its monomer of chitin E. So, of course, it is a marker of fungal necromass. Next one, please. The paper with the new method on LCMS is already published here. Next one, please. And basically, we do not have any DNA, RNA methodology for the necromass because DNA and RNA would decompose relatively quite fast after the death. So that seems to be the only choice right now for studying microbial necromass. Next one, please. Yes, next one. Yes, so melanin concentration. Probably you've heard already that melanin is assumed to be one of the very recalcitrant products of fungi, and that it is some sort of a polymer which is present in the cell walls and which is potentially highly persistent in the soil. So maybe that melanin from fungal necromass is especially responsible for slow decomposition of fungal necromass. Next one, please. Here you can see that people are usually using the term melanin, but it's far more complicated because there is at least 4 different types of melanin produced by fungi. Here are the most typical ones. On the left, you can see pheomelanin, not very typical for the fungi, but still some fungi can produce it. As you can see, they are also, this pheomelanin is also containing nitrogen and sulfur, so not very economically good version of melanin because fungi has to spend a lot of nitrogen and sulfur for that. In the middle, you have allomelanin. This one is very typical for most of the fungi. It does not contain any nitrogen and sulfur. And on the right, we have eumelanin. Some fungi can still produce it. It contains nitrogen. And as a matter of fact, eumelanin is that melanin which we are having in our skin. Next one, please. But we do not have any method to measure exactly the melanin concentration in the soil. On the basis of DNA, we could tell that, for example, fungi A can produce quite a lot because it has that information in DNA. But we do not have the method to really take the soil sample and tell that it is 2 grams of melanin. Next one. Okay, that one I've told you already. Next one. So we figured out some method on LCMS to measure it. In the middle, that red ring in the middle of the chromatogram is showing you 2 peaks of eumelanin, and on the right, 2 peaks of allomelanin. It is typical soil from Finnish forest on mineral soil. So as you can see, we were able to build up the method able to quantify concentration of these 2 types of melanin and also to find the difference between these 2 types of the melanin. Next one, please. Yes, so another method which we developed during last couple of years is DNA methylation method. Typically, metagenomic protocols do not capture the methylated DNA. Next one. So methylated DNA, so there is a methyl group bound to cytosine, which is blocking the replication and so on. So it's good to know that kind of a level of DNA methylation because it's also a marker of the stress. So we figured out that kind of a method using HPLC with mass spectrometry. On the right, you can see 5 peaks. Next one. So there is a cytosine, methylated cytosine, the second peak, and the rest of the typical DNA things, adenosine, guanosine, and thymidine. Next one, please. So to summarize the first part of my talk, that methodological part, in HoliSoils we developed the following methods. DNA methylation, so we can get information about % of methylated DNA. A precise method for fungal biomass concentration. We've started to fight with dividing fungal biomass into different functional groups. So especially we are working on that arbuscular mycorrhizal biomass. We also developed the method for microbial necromass concentration measurement and melanin concentration. Next one, please. So jointly, I think that these methods can really support DNA, RNA analysis, and together we can get better insight into soil processes. Next one, please. So in part 2, I will tell you something more about the mechanism of carbon stabilization. I picked a couple of them. I will tell you why these. Next one, please. But at first, I wanted to underline what is the carbon stabilization because people are using that term not always, I think, correctly. So all the processes or mechanisms leading to prolonged turnover times of SOC, of soil organic carbon, can be called carbon stabilization. Next one. But turnover time of stabilized organic carbon cannot be given as absolute values because sometimes people are telling me that this pool of carbon will be stable for 100 years. Well, probably not exactly because it depends on the environmental conditions and soil properties. Next one. And yeah, so yes, and next one. So here on the left, you have one more time underlining the importance of microbial necromass. And of course, this process of carbon stabilization is under influence of many factors. In the middle, you have a very long list, like, of course, environmental conditions, temperature, moisture, and so on. Biodiversity, not only of microbes, but also plants, root depth, root chemistry, priming, microbial biomass, community structure, soil fauna, necromass, amount of chemistry, necromass thickness, and of course, interactions with minerals, taking into account amount of them and also chemistry of minerals. Next one, please. So here I wanted to underline one more time the role of microbes inside the soil. Next one. Quite often we are having some kind of a balance between stabilization and decomposition. Quite often we are thinking that microbes can only decompose, but also, like I mentioned before, they are also able to stabilize. Next one. And next one. So basically, when we are thinking about carbon stabilization, we care to some extent about these living guys, living microbes, but especially we are caring about these, next one, dead ones. Next one, please. And next one. So here's one kind of interesting example of recently found mechanisms. It seems to be that one specific fungi, which is ectomycorrhizal, it is called Cortinarius. It is having a good tendency to decompose organic matter, and on the basis of studies done in Sweden, in many places, it seems to be that if there is more Cortinarius in the soil, there is less organic matter concentration, which means that this fungal species is so good in decomposing that it affects the organic matter decomposition so strongly. Next one. This I also like very much. It was published a couple of years ago, and it takes into account, let's say, the lifestyle to some extent of microbes. That some of them, small part, they are very active. They are, to some extent, you can tell that they are doing the job. But most of them are just lazy and sitting for the ready food. So this could be also taken into account. Of course, it makes it really complicated because in that way, fungal biomass or bacterial biomass itself does not mean much, but it's important who is doing the job. So here probably RNA would really help. Next one. Yes, here I wanted to show you the mechanism which I managed to study and publish still before the HoliSoils, but we worked on it a bit more inside the HoliSoils, because quite often we are thinking about litter as a separate chemicals. Okay, something is going to the soil and it will stay there forever. So maybe not forever, but so for some longer time. But here I wanted to underline that these things can interact with each other. Because when we are thinking about plant litter and recalcitrant structures, we are thinking about lignin, that really big structure on the left, or tannins, this smaller one on the right. Next one. And when we are thinking about fungi, we are quite often thinking about, next one. In the middle, it's glucosamine dimer, so chitin and all these melanin types about which I was talking about before. So in practice, if you would add most of these compounds, maybe not about melanin, but chitin and lignin and tannins to the soil, well, they will decompose quite fast. So why they are quite often found in these more recalcitrant pools? Next one. Yeah, so it's important about interaction. Next one. So like I was underlining, most of the carbon in the soil is coming from fungi and roots, and organic nitrogen pool contains mainly peptides and amino sugars, and fungal necromass seems to be seems to form very stable organic matter, but as you know, fungal necromass contains a lot of nitrogen, so why not to eat it at first? So how it's possible that the things which are separately decomposing relatively fast, they can form something more stable? Next one. So after a longer moment of thinking, next one, we figured out that maybe they are just simply interacting with each other and making the complexes, which are creating more stable organic carbon. Next one. And next one. We went through a couple of experiments. We started from the lab experiment in which we have had, you can see that small, nice pine with a lot of ectomycorrhiza, and we fed it with fungal necromass labeled with 15N or with complexes with fungal necromass. And you can see that 15N was taken up far faster from pure fungal necromass without complexes. Next one. It was visible in roots, stem, needles, and the whole seedling. So the same pattern. Next one. We also went into field experiment in which we've added fungal necromass to mesh bags. And this fungal necromass from the mesh bags was to some extent decomposed slower when the roots were able to reach it and probably build up the complexes. Next one. Here you can see that there is a nice correlation between a stable soil organic matter. Here I focused mainly about nitrogen and condensed tannins. Next one. Yeah, so it's harder to decompose fungal necromass if it is in complexes with condensed tannins. Next one. And next one. We also did another field experiment in which we've added two mesh bags of different sizes. You can see the sizes here. So 1 millimeter from the left, so the roots were able to reach it, 50 microliters and 1 micrometers, and 1 micrometer. Next one. We added there the soil, and we wanted to take a look where soil organic matter can be built up or decomposed faster. Yes, next one. So in 1 millimeter, of course, there was the biggest amount of bound tannins after incubation, especially after 3 years of incubation. Next one. So just to underline what I was telling last time, so from the lab experiment to field experiments, we proved that interaction between different compounds coming from fungal necromass and plant litter can make the organic carbon more stable. Here's another thing about minerals. Quite often we believe that mineral-associated organic matter is really strong. It's really hard to decompose it. It's really hard to remove organic matter from minerals. Next one. But we know also that roots and also microbes can exude these two small organic acids, citric acid and oxalic acid. And as a matter of fact, these acids can quite easily remove some of organic matter from minerals. Next one. To check how does it work, some years ago, we added, next one, to the soil separately BSA, so protein. So of course it was decomposed very fast. We also added that BSA, so protein, in complexes with condensed tannins, and we observed that, next one, you can extract far more BSA from the soil if it was added separately. So at first protein and later condensed tannins, but when you are adding complexes together, it's really hard to remove it from the soil. So that interaction is also increasing the interaction with minerals. Next one, please. Yeah, like I said, yes. Next one. In last part, I will show you the case study of Karstula, in which we followed the effect of nitrogen fertilization on soil organic matter stabilization using combination of different methods. Next one. So the Karstula site is, the experiment was established in 1959, so that's really big plus of that experiment. It means that it's really long term. The typical forest there is, yes, it's Pinus sylvestris is the most common tree there. Next one. And here you can see where it is exactly in Finland. Next one. So we build up on the basis of literature and what we assumed, following 4 potential hypotheses, because quite often in papers you can find that nitrogen fertilization is increasing carbon content in the soil, so carbon stock is higher. But why? The first hypothesis is that nitrogen fertilization can change the pH, but usually in practice in the forest there is also liming, so it's rather not possible. The second hypothesis is that that addition of nitrogen can switch fungal community in favor of fungi unable to degrade polyphenolics. So we have more polyphenolics at some point, and maybe because of that we have more organic carbon, because these polyphenolics can nicely interact with organic matter. The third hypothesis is that it's simply more plant litter, so more litter come into the soil, so there will be more fungal biomass and more fungal necromass to stabilize the carbon. And last but not least, nitrogen fertilization could potentially also change ground vegetation, affecting all those organic matter processes. Next one. So here are our results. So we've got, please take a look into these green columns especially. We've got more organic carbon and also organic carbon stocks after nitrogen fertilization. Next one. And next one. In case of enzymes, we've got also basically almost always higher enzymatic activities under nitrogen fertilization. Next one. Especially in organic layer of the soil, less in mineral layer. Next one. And next one. We've got also a somewhat higher amount of ammonium in the soil after nitrogen fertilization, which is kind of logical. Next one. This was a bit surprising for me in the beginning, because I was thinking that maybe also the amount of tannins in the soil should be higher because we have more litter, but it was in opposite way, as a matter of fact. Under nitrogen fertilization, the amount of tannins decreased. So potentially this could be against the hypothesis that nitrogen fertilization is increasing organic matter content. And also, it has also another negative background, let's say, that it can also cause lower resilience of the forest to, for example, beetle outbreaks. Next one. And next one. We found also under nitrogen fertilization far more ergosterol, so fungal biomass, and more chitin, so fungal necromass. Next one. And next one. Even there was more fungal necromass in mineral layer under nitrogen fertilization, so the effect was really strong. Next one. And next one. And next one. So to summarize, let's say, our results. Next one. You have from the left parameter which we've studied, in the middle the effect on control, and on the right the effect on nitrogen fertilization plots. As you can see, all of these things were higher. I mean, tree growth, litter fall, soil organic carbon stock, stable carbon, mineral associated organic matter, enzymatic activities, bio and necromasses. Next one. But the two things were about to be the same. So greenhouse gases production and microbial community structure. Next one. But there was one negative result that tannins were lower under nitrogen fertilization. But taking all of these results into account, we could basically suggest that nitrogen fertilization, at least in this dose which we used, because quite often people are using in their experiments far higher doses of nitrogen fertilization. But if you are not going too much up with nitrogen fertilization, you can basically build up more soil organic carbon. Next one. So it is just a conclusion about which I was talking about. So the main mechanism was just increased plant litter and fungal biomass, and which is later stabilized as a necromass in the soil. And these things, at least in this specific site, which was really nitrogen limited, it was really working to increase organic carbon after many years of the experiment. Next one. Yes, here I wanted to underline just why we more like in different way, that by combination of the different methods, so greenhouse gases, metagenomics and biochemistry, we can support our hypothesis and build up some more in-depth understanding of the processes. So I would really recommend to combine in your studies greenhouse gases measurements, metagenomics, metatranscriptomics, chemistry, and modeling, and all the time development. And by combination of these powers, next one, you can build up, next one, Captain Planet, I mean organic matter processes understanding. Next one. So thank you, that's all from my side. In case of questions, I see that there are some questions probably in chat, but you can also write me an email. Next one. You can find all these methodological papers and other papers here. And next one. And last but not least, as HoliSoils is finishing, I would like to underline that I'm really grateful for being a part of this crew, and thanks for all of the members of HoliSoils family. Thank you.
Thank you, thank you, Bartek, for excellent talk. And as you have noticed, this presentation and webinar is recorded, and if you didn't catch all the details, you can listen it again and share with your colleagues. But now we have time for questions. I have been following that we don't have any questions there in the question answers box. We have about we have 40 participants online and almost 10 here in this room altogether. So we can also have questions from this room, and I will follow what we will have in question answers box in there. Peter.
There is one question from the room, and this is about the potential of using 24-methyl ergosterol in AM fungi. There is also the method using NLFA, non-lipid fatty acids for AM fungi. Would you somehow compare the potential of these two methods? Why not to use the NLFA? What can be the advantage of this 24-methyl ergosterol?
Yeah, NLFA, it's yes, it has some markers for arbuscular mycorrhiza, but some of these markers were also in some papers shown as a maybe not exactly coming from arbuscular mycorrhiza. It can be exactly the same with 24-methyl cholesterol, because it seems to be from some papers that some arbuscular mycorrhiza can produce still a bit different version of ergosterol. So not 24-methyl cholesterol, but still something else. It is just something what I have to work more on that. And the plus of the HPLC method would be that it's coming from the same method, exactly. In the same run, you have ergosterol, and using different wavelength, you have also the result for 24-methyl cholesterol. And that's exactly the same extractions from exactly the same sample. And it's pretty fast comparing to an LFA, definitely.
Thank you, Bartek. Any other questions from the room? We don't have any in the chat.
Maybe I could ask about you showed that tannins were kind of reduced relative to control in karstula, and you were saying that this can affect the resilience. Why is it tannins, are there less tannins because of somehow there is more easily nitrogen available, or is it like why do you see that? Because trees need to defend themselves in future as well, and if you grow more faster, maybe you're more prone for disturbances.
Yeah.
So
there was a theory which was let's say bombarded later, but still it works here pretty well. It was carbon nutrient balance theory. So if you have enough nutrients, you are growing fast and you don't care about secondary metabolites because you are growing so fast that there is no carbon to feed it. But in case of stress, if you are having low level of nutrients, the organism, the tree is under the stress. So it produces also secondary metabolites. So that was my only explanation, because of course, otherwise that's not really smart to have a lower immunity system, let's say, of the whole ecosystem.
Right.
But yes, but last thing, sorry. But quite often this mechanism of reaction to abiotic or biotic stress, whatever, to every stress, are going through very similar metabolic routes. So lack of nutrients is also causing it's activating these metabolic routes, which are increasing the production of secondary metabolites, including tannins. I haven't checked the other metabolites. This would be also cool to check if the whole metabolism is switched in that direction.
Interesting.
Yes, thank you. As far as I know, there is no questions in questions and answers tool. So this means that this was crystal clear presentation. Thank you, Bartosz, for that. But we still have 3 more minutes to make questions if we want to keep the schedule. I will use the last 20 seconds for some advertisement, but you're still free to make questions if you have any. Since we don't have questions, I try to show you something from my screen. But for us, this Zoom doesn't really work. I don't mind. I cannot show anything from here from you. But I was meaning to advertise the coming following webinars of the HoliSoil. We still have sessions 4 agreed for October, and next one will be about monitoring of mineral forest soil. So the following ones will be more focused on training of the experts who might be interested in soil monitoring methodologies and our experience in HoliSoil. And October 8th at 2 o'clock Central European Time, we will have the next one. You will find full information from HoliSoil website, which is holisoil.eu. In addition to these webinars, which are online and where we have guest speakers, we are also organizing a policy event in Brussels and online. It will take place in October 10, and you're welcome to join. It's from 9:30 to noon about forest soil for climate smart future, the relevance of soil in policy and practice for forest management in Europe. And we will, for instance, show our results on scenario analysis, what applies for whole of Europe this is kind of coming activities of the HoliSoils project. We are there in LinkedIn and Blue Sky, and you can follow us in our website. And there is lots of scientific publication coming from our team members. So hopefully you will enjoy reading them. Any comments? I will check once again whether we have some. Yeah, not questions so far. If you have any specific questions to Bartek, as he told, you can send an email and contact him later. And the information about future seminars is shared also here in the chat box of this webinar. So welcome and see you again. And thank you for joining today.
We have one question, Raisa, before finishing the webinar. We have one.
You.
Yes.
Thank you. Yes, we have here a question. First, thanks. And then I was wondering if you developed methods, if your developed methods could be combined with isotope tracing, carbon or oxygen. Are you ready to respond on that, Bartek?
Yes, basically, I've been already a bit working on it. Also, basically, that 13C, I do not have ERMS in that system which I'm using, but still, of course, 13C is heavier than 12C, so I can see the isotopic pattern also to some extent there. So at some point, yes, definitely, it's possible to develop it like this. I was thinking that I could try to separate the signal from fungal biomass of ectomycorrhiza, as they are having potentially different isotopic pattern because they are supported with from the plant with sugars, right? So probably it's possible.
Yes, thank you. And in our team, we have some earlier publications which were using the isotope signals of different fungal species, and then we were more focusing on dead wood and the composition of that substrate. But anyhow, that can be combined. And thank you, Maya, for this question. If not any further questions at the moment, as I told, we are ready to close this session, and thank you for joining, and hope to see you next time online. Thanks.
Thank you.